Now what happens when I look back in the question, I should say, is that I know that these two genes are 12 map units apart, which means 12% of the time you're going to get a crossover between them, because that's the definition of map units, right? Map units and percent recombination are the same thing. So 12% of the time I will get a crossover, which means 12% of the time I will get non-parental genotypes. So added together, these would happen 12% of the time. So I know that 6% of the time I would get that, and 6% of the time I will get the other non-parental. So 88% of the time I will get parental, so 44 of that one, and 44 of that one. So now I know what a non-parental genotype looks like, and I know the percentage amount of time it's going to occur. I also know that 100% of the time in this cross, the other one is going to be little a, little b. And this question actually asks you what percent of the offspring are going to be each of the 4 possible genotypes. So now all I have to do is write down all the possible combinations here, which are just 4, because basically each one of these chromosomes is going to get together with this one. So if I had to look at the F2 here, I get big A, big B, little a, little b. So I get essentially this guy. Oh no, oh no. Gonna run out of room. So I get essentially take this chromosome from one parent, this from the other. I take this little a and little b from one parent and one from the other. I take big A, little b from one parent. And of course, the other one's always going to be this. And last but not least, I take little a, big B from that parent. And of course, the other is like that. And I know the percentage of all of these things, right? So the first two, this is going to happen 44% of the time. This is also going to happen 44% of the time. This is going to happen 6%, and this is going to happen 6%. If I was to write these down the old school way, it would be this, it would be this, it would be this, and it would be this. And that's it, right? So using this scheme, I can figure out exactly the percentages of everything. I think this question is useful because if I now show you, so does that make sense before I move on? So this is basically for A. And I think this question is useful because it shows you how confusing this can be if you don't draw out the chromosomes. Because in 4B, as you will see, things look different. So in 4B, what you're asked is, would these numbers of the F2 be any different if the parental generation had been this? And the reason that I think this question is useful is because if you got these two organisms together, right, you get this. Actually, right over here. So the F1 is going to be a dihybrid, just like it was previously, right? So if we look at the first scheme up here, the F1 is a dihybrid, just like it is here. So it looks exactly the same if you write it the old school way. But what you'll see is that when we introduce this new way of writing across using the chromosomes, these guys are, of course, pure breeding. So they look like this. We'll take one chromosome from each parent. So we get one from this guy and one from this guy. Of course, if I was to write this down the old school way, like I said, it looks just like this. But look at the difference now. Up here, the alleles are arranged differently. Even though it's the same genotype, the alleles are on different chromosomes. And that's key to figuring out the rest of this question, right? Because now the F1 is a test cross. So if I was to write out the gametes, again, I always start off by writing the parental ones because it's just easier to group them together that way. So one of the parental ones is that. The other one is like that. One of the recombinants would be little a, little b, because the crossover would occur here. And the fourth one would be, of course, big A, big B. And the only possible gamete on this side is, of course, that. Now, in this case, these guys are parental. And these guys are non-parental. And of course, we're told that these genes are 12 map units apart. So in this case, this is going to happen 44% of the time, as is this. And I've calculated that these guys are going to happen 6% of the time. So now what you see is if we look at the possibilities for the F1, F2 generation, we're going to do the same thing that we did before. Of course, all the bottom chromosomes come from the homozygous recessive guy. And then I just write down the ones from the top. So little a, big B, little a, little b, and finally that guy. So and of course, from up here, I know the percentage times this is going to occur. And 6 and 6. And what you see, because we flipped those alleles in between those two different genes, we've basically flipped the numbers of recombinants and non-recombinants. So if you compare this to above, for example, if we look at the homozygous recessive guy, that was 6%, and up here it was 44%. Because in this second scheme, this is the homozygous recessive is a recombinant genotype, and at the top it was a non-recombinant. So I like this question because it specifically gives you, it shows you very specifically how these things work out. So that's how I approach a linkage question, by drawing out this scheme. Please notice that with these kind of questions, you're sometimes asked essentially to do this question backwards. So I'm not going to go through the entire question because I just don't have enough time to do that with you guys. But take a look at the very last question in this question set. This was these. When was this? Oh, corn. This is the Z-Maze question. So this is the corn question, where you're shown two different numbers of offspring from two different types of crosses, and you're asked to work out the recombination the recombination frequency and all that kind of stuff. With that kind of question, what you're basically doing is this in reverse. So for this one, you're basically gonna, you're asked to do, for question 8, you're asked to do a statistical test to see if these are segregating independently or not. What you're looking at is a test cross in this case. It's very easy to figure that out because you're told what the dominant and recessive phenotypes are. So you know that the F1 cross is a dihybrid crossed to a homozygote recessive, just like the F1 in this case that we looked at. Remember that a test cross in a non-linked situation always gives you a 1-to-1-to-1-to-1 ratio. That's clearly not the case when you look at these numbers. So that what we know here is that these genes must be linked. And quick word to the wise when you're looking at linkage questions, whenever you see these numbers, the lower numbers are always the recombinant phenotypes, because by definition, the recombinant stuff happens less than 50% of the time. So the lower numbers are always the recombinant phenotypes and recombinant genotypes. It has to be that way. And if you look at those two crosses in number 8, you will see that basically you're looking at this same question just with numbers instead of, you're given the numbers and you have to figure out the percentage these things are recombining at to figure out the recombination frequency, which gives you the map units because they're the same thing. And you're basically looking at the same situation where you've flipped alleles in one cross versus the other cross. One last thing about that question before we move on. The very last part of question 8, it tells you to merge the two sets of progeny data. Sometimes that's confusing for students because you're like, how on earth do I figure out how to do that? But remember that what you're looking at is you're just calculating a percentage, and all you need to work out is the percentage of recombinant individuals in that experiment. Like I said, the recombinant individuals are always the smaller numbers. So if I was merging those two data sets, I would say to myself, okay, first of all, how many progeny are there in total? There's 2,260 in the first table. There's 845 in the second. So I'll add those together. That's what merging the data sets is, right? To get my 3,000 and whatever it is. And then I just need to figure out the total of recombinant individuals and the total of non-recombinant individuals. And I know that the smaller numbers are always recombinant and the bigger numbers are always non-recombinant. And so I'll just add all the big numbers together, add all the small numbers together, work out the percentage that are recombinant, and that gives me my, percent recombination frequency, which is the same as map units. And it's about 27% or 27 map units in this case. So my advice for linkage questions is to basically always draw a scheme where you can see the chromosomes and you can see the alleles on those chromosomes so that you can figure out exactly what a non-recombinant or parental chromosome looks like, and also what a non-recombinant one looks like. Does anybody have any linkage questions? Okay. So last but not least, I want to talk about the final assignment that you guys are finishing up, which is assignment number 11, gene regulation in bacteria. And specifically, what I want to talk about is question number 8. All right, so question number 8 is this crazy looking table that you were tasked with filling out, right? And again, I want to show you a sort of trick or a strategy for how to approach this question, which is going to make it a lot easier to figure out what's going on. When I'm tasked with a question about operons, like the Lac Operon in this case, what I like to do is draw a little picture of the Lac Operon to help me out so I can figure out what's going on in these different partial heterozygotes, right? So what I'm going to draw is the Lac Operon and also the Lac Repressor gene, which is not technically part of the operon, but is really important for how it functions. So if I was to draw out the Lac Operon, right, I would draw something that looks like this. And I'm going to draw it, and I'm going to draw it this way. So first of all, I'm going to draw, and this is one piece of DNA, right? I'm going to draw, oh, that's not how you spell lac at all. I'm going to draw this gene, which is the Lac Repressor, right? And then I'm going to draw this little part of the Lac Operon. So the promoter and the operator. And then for the purposes of this question, right, I just need to draw the two genes, Lac Z and Lac Y. Okay, so that's a version of the Lac Operon, right? Notice that when I draw this, and this, so all of these letters represent the letters in that table, right? So if you look at the example genotype on the very first row of that table, it's IPOZY. Those represent these letters, I, P, O, Z, and Y. Notice that there's 3 genes involved in this scheme. Technically, this part is the Lac Operon. And this guy is the Lac Repressor. Annoyingly, the Lac Repressor is called Lac I. That's because it was originally called the inhibitor, which is annoying, but now it's called the Lac Repressor, which is a better name for it. But unfortunately, in science, the first name always sticks. So there's only 3 genes here that are actually going to make proteins, right? So this Lac I is going to make a protein, which is the Lac Repressor. This is going to make a protein, Lac Z. And this is going to make a protein, which is called Lac Y, right? And remember what these guys do. So Lac Y is permease, which allows lactose into the cell. And Lac Z is beta-galactosidase, which allows the cell to break that lactose down to use it as an energy source. So in the Lac Operon, there's these 2 structural genes that are important. There's, of course, another one that I didn't draw here because it's not functionally important. And then there's this repressor that's made by Lac I. Lac I is constitutively expressed, so there's no regulation. So this is called and that's really important. So that's not regulated in any way, shape, or form. It's just made all the time. And of course, Lac I is key to understanding how this operon works. So when there's no lactose around, Lac I will bind to the operator. So it's a protein, it will physically bind right here. So Lac I makes this the Lac I gene makes the Lac I protein, which is going to float around in the cell and bind to the operator. Remember, the function of the promoter is to bind this really important protein known as RNA polymerase. And RNA polymerase, its job is to make Lac Z and Lac Y. In order to do that, of course, it has to move down this piece of DNA and transcribe those 2 genes into mRNA, which will then be made into protein. When Lac I, when the repressor is bound to the operator, as you can see from this scheme, it's like physically in the way. RNA polymerase cannot move down this piece of DNA because it's physically blocked by this Lac repressor. So it's like a roadblock to this happening. Of course, in the presence of lactose, the inhibitor falls off the operator, so the repressor drops off, allowing RNA polymerase to make these Z and Y proteins, allowing the cell to use beta-galactosidase. So this is the wild type situation, right? When I'm approaching one of these questions, in normal bacterial cells, there's just one version of the Lac Operon. When this has been studied experimentally, what you do is you take an E. coli cell that has one version of this Lac Operon in it. So here is the same thing I just drew above. So that's like the Lac Operon in an E. coli cell. And then when you're trying to figure out how these different things work, you actually introduce a second one. And that's what these partial heterozygotes are. So what you're looking at in the little table there, when you get 2 lines, is an E. coli that contains 2 pieces of DNA that contain the same kind of genes. So this scheme that I've drawn is basically representing in the table this kind of thing. right? So that's what's written in the table. When I'm approaching this question, what I actually like to tell students to do is to draw these two versions of the Lac Operon, because that's going to help you figure out what's going on. And the reason it's going to help you figure out what's going on is that, as we know, right, this gene I here, is going to make a protein. Now, this protein could be made by this version of the gene, or it could be made by this version of the gene. Both of these versions of I are going to make the repressor protein, which is just going to be floating around in the cell, right? Like lots and lots of copies of it. This protein, once it is made, it's free to go wherever it wants, right? So this repressor protein, all it wants to do is find a DNA sequence that it recognizes, and it wants to bind there. So this protein could bind to this operator, no problemo, and it could also bind to this one. It doesn't care. It's just a protein that's looking for a place to go. So this protein, I, geneticists call this a transacting factor. And it's just called that because it literally means it can sort of transfer itself and go anywhere you want, right? This is the key to understanding this problem. This is a transacting factor. It can float around and go wherever it wants. Things like P and O, so this promoter and this operator for this top piece of DNA, these guys can only affect, so this transacting factor can regulate this piece of DNA or this one, doesn't matter, because it can just float around and go wherever it wants. If you look at the promoter and the operator, however, they would be called cis-acting. What that means is they can only regulate stuff that is on the same DNA strand as they are. So this promoter on this top piece of DNA can only affect this gene that's right downstream of it. Likewise, this promoter from the one below can only act on the genes that are downstream of it, right? The promoter on the top strand of DNA doesn't interact with this bottom strand at all. It is cis-acting. I is a transacting factor. So it's normally true that transacting factors are proteins that float around and do stuff. Cis-acting factors are regulatory pieces of DNA. And that is key to understanding this question. And I'll go through an example to show you why, and then you guys will be glad to hear we can conclude. So let's do C. So if you look at C, this is what the question looks like, right? It looks like this. I, P, O, Z, Y. And then the other version is I, P, O, Z, Y. And then on this top version, if you look at part C, we have plus, 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 minus, plus. And then on the bottom one, we have I plus, P minus, O plus, Z plus, and Y minus. All right, so for this particular example, right, like I said, I start off with as soon as I see this problem, I go, okay, and draw my little scheme so I can figure out what's going on, because there are two versions of this operon in this cell. There is one, here is the other. And of course, if I was doing this in an exam, I'd probably just draw 2 of these and then, that's enough. I wouldn't draw 1 for each different thing. Because you can just scribble out what you need to. All right, so what do I know about these versions? I know that, so plus is wild type, so functional. Minus is mutant, non-functional. So for this top version, everything is functional apart from this structural gene Z. So I know that Z does not work for that top version of this, this operon. If I look at the bottom version, look, there's a mutant Y, so that doesn't work. Fair enough. I look for any other mutants. Oh, this is a big one. So P is mutant. This promoter for this bottom one is dead. What does that mean? That means RNA polymerase cannot bind to that promoter. That is really important to know. Because now when I'm looking at this cell, I say to myself, well, this promoter's dead. So this whole operon, I don't care about it because the promoter's dead, can't do anything. This operon is not going to work at all. The I gene here is still functional. So it's, and this I gene is functional because those are both pluses. So there's lots of repressor being made in this guy. But I know that in that bottom operon, you're never going to make this Z, even though it's a wild type gene, because there's no promoter. So I don't have to worry about that. If I look at the top one now, I see that there's a functional promoter, there's a functional operator. So this is going to be regulated just like the wild type. In other words, if lactose is present, it's going to be on. If lactose is absent, it's going to be off. But it has a mutated structural gene. So in the presence of lactose, this is going to make Y. So the permease is going to be made in the presence of lactose, but not in the absence of lactose because the repressor will be bound. So I can add that to my table, right? So if you look at C and you look at the 4 columns, if you look at the 2, or the first 4 columns, if you look at the 2 on the right, we're looking at the permease enzyme, the Lac I Gene product. If lactose is present, I'm going to put a plus in that column because I know it's going to be made. If lactose is absent, I will put a minus in that column because just like the wild type, it will be repressed in the absence of lactose. However, for Lac Z, this guy is never going to make Lac Z because it has a mutation in the Lac Z gene. This guy has a Lac Z gene, but it's never going to be made because it's got a defunct promoter. So for this version of the table, Lac Z is going to be minus and minus because dead gene, dead promoter. Lac Y is going to be plus minus, right? Because made in the presence of lactose, not in the absence of lactose. And so I think this kind of scheme that you can draw is really useful for figuring this out, right? Because really what you're trying to do in these problems is figure out what is made and when, because the cell as a whole, whether or not it's going to be able to metabolize lactose, essentially depends on whether or not you're making lactose in the presence, you're making these genes in the presence of lactose. Because in order for this cell to use lactose as a energy source, it has to both be able to import it into the cell with Lac Y and digest it with Lac Z. And so this individual cell right here in part C, it makes permease in the presence of lactose. So lactose could get into the cell, but when lactose gets into the cell, there's no Lac Z to be able to digest it. So this individual will be Lac minus because it cannot use lactose as an energy source. And so using this scheme, I think you can essentially go through each one of these different genotypes and use this little scheme to say, okay, what operon is making what under what circumstances, and then once you figured that out, you can say whether the cell is Lac plus or Lac minus. There's a couple of other genotypes that are involved, a couple of other mutations that are involved in this problem. For example, there is the OC one, which is a constitutively active operator. What does that mean? It means the operator can't bind any type of repressor at all, which means anything downstream, the Z or the Y, of an OC mutant will always be made because it's unregulated. It'll be made in the presence of lactose or in the absence of lactose because the repressor cannot bind to an OC operator. So using that logic and this little scheme, I think you can figure it out pretty easily because all you basically have to do is understand that, you know, promoters and operators are cis-acting. They only act downstream of themselves, whereas things like the repressor is transacting. It doesn't care. It can bind to any one of these operators that it wants to. So it'll bind to both of these operators no matter what. So if you have one mutant repressor and one wild type repressor, then the repressor will be made and it will bind to both operators because it's transacting. Yeah, and that's how you address one of those questions. So with that question, if I was presented with that question in an exam, I would draw these two schemes and then just go through it line by line and ask myself, okay, is Lac Z made in the presence of, sorry, are these guys made in the presence of lactose or the absence? And you can figure it out by, figuring out where this repressor goes and whether or not you have mutant promoters or operators. If you ever see a dead promoter, it's super easy because you just discount that version of the operon entirely because it's dead. So those ones are a little more simple. Okay, that's what I have for you guys. So does anybody have any other questions for any other stuff? I think that as far as problems go in the exam, they're gonna, if you know these schemes to work stuff out, then you should be able to address each one of those problem types with at least a solid strategy. And then if you follow those strategies, you can kind of figure everything out around those strategies. So you see it's a linkage problem, you draw the chromosomes. If you see that it is a Lac Operon problem, you draw your Lac Operons. If you see that it is an epistasis problem, you draw your little epistasis cheat sheet for 9331. And using those approaches, hopefully it'll be okay. So, so for the exam, I would definitely have a notepad and paper with you so that you can write these things down, these little cheat sheets down in order to get to the right answer. So does anybody have any questions?
I kind of wanted to ask a question about number 9 on the gene regulation in bacteria.
Yeah, sure.
So looking at the table, strain 3 and strain 6, they are they all have active enzymes, whether love is found or not.
Yeah.
And so for me, like that points to a problem in either the operator or the repressor, but I don't know how to differentiate which one is which because they would both result in active enzymes.
Absolutely, yeah, exactly. So, so for you guys that are not that familiar with this question, what Jen's talking about is basically this is a question that's sort of similar to the previous one, but you have to figure out which thing is which. So, for example, in this case, we have these different letters that represent things like the operator, the promoter, the repressor. So we have V, W, X, Y, and Z. Now, as I told you guys, dead promoters mean dead operons. So if we look at a mutant where nothing ever happens, we know that that must be a dead promoter. So that's like number 4 here. So if you look at number 4, you have N's across the board, which tells me that operon is dead. And that is because of a mutant W, which means that must be the promoter. Likewise, we have 2, you're told in this problem that just like the Lac Operon, there are 2 structural genes. So if we ever see one gene affected, one enzyme affected and not the other one, we know we're talking about a structural gene. You see that in the very first, not the first one. You see that in...
Sorry to interrupt, but I think X is the...
Oh, sorry, we were wrong. Oh yeah, sorry, I was looking at the wrong line. Yeah, that's X, sorry. Thank you. So X is the one that's dead across the board. It's my old eyes not being able to read the tiny things. Thanks, Jen. So if you see where one enzyme is affected and not the other one, so you see that in number 2. So V must be enzyme 2. So that's the logic that we're using for this. And the problem with this, question is that when you look at number 3, for example, you get active enzyme all the time. So that is either a mutant repressor but could also be a mutant operator. Because that has, those kind of mutations would have the same phenotype, right? You mutate the repressor, the operon's on all the time. You mutate the operator, the repressor can't bind to it, it's on all the time. And you actually see that in strain 3 and also in strain 6, which is Z. So that could also be this. And the way to figure out which one is which is to look at the very last experiment, which is number 11 on the table. And what you see in number 11 is this situation. So number 11 is actually the combination of 2 operons. So we have, the bottom one is all wild type. And the top one is just mutant in Z. So V plus W plus X plus Y plus, but Z minus. And this is going to be how we solve this problem. And basically, the way to solve the problem is to understand that the repressor can move around. So it can, it's a transacting factor, and the operator is not. So what you see when you look at the phenotype for that experiment, right, is that both enzymes are active all of the time. What would you expect if Z was the repressor? So if Z was the repressor, you would ask yourself, does this cell make any repressor at all? And the answer is it would, right? Because you have a mutant repressor, but hey, here's a regular one. So if Z is the repressor, you would expect the repressor to be made by this operon, and that repressor would regulate both of these guys because it's a transacting factor, right? So if the repressor, if the wild type repressor was present in this cell, would you expect the operon to be regulated normally if there was active repressor floating around in this cell?
Yes.
Exactly, because it could repress either one, right? Do you see that? So is this, in experiment number 11, does it look like the wild type in strain number 1?
No.
It doesn't. So that tells me that Z cannot be the repressor. It would have to be the operator. Now, if Z is a mutant operator that can't bind the repressor, what would you expect that operon to do if it cannot be repressed? Would you expect it to be active all the time if it can't be repressed?
No. It would be inducible.
But remember, it can't be repressed, right? If it doesn't have, so think about it in terms of the scheme, right, up here. So if you have an operator that cannot bind repressor, then there's nothing to get in the way of RNA polymerase, right, if repressor can never, ever bind. And so it's then unregulated. It just is made all the time because there's nothing to stop it being made. There's no repressor. So if you have, does that kind of make sense? So if you have two versions of an operon, and one of them, so if I had two versions of the Lac Operon, for example, or if I had, yeah, okay. So if I had two versions of the Lac Operon, I don't even know if this is a situation in question 8, but if I had two versions of this, right? And one of these is OC. So this guy, it's called OC because it's constitutively active. Which means this and this are going to be made all the time, no matter what. Right? So in this case, if I ever see an example of something that's constitutively active, and these are normal, these are wild type, then this cell is going to make these all the time, no matter what, because it's not regulated anymore. So even if, for example, this had a negative promoter, which totally killed this operon, a cell that had both of these things would just make these all the time. So it doesn't matter what's going on up here, even though this is completely dead, this one is active 100% of the time. So, and remember that an operator that's active all the time is exactly the same phenotypically as something that lacks repressor, right? So if you look at number 11 in this strain list, you see that it's active all the time. it makes active enzyme 100% of the time, which means that Z has to be the operator and W has to be the repressor, because there's no other way to get that. Does that kind of make sense?
Yeah, I had actually realized that I forgot to copy and paste 9 through 11 on my Google Docs. So
Oh, no, go ahead with that.
why I could not see that.
Ah, gotcha. So yeah, that's your problem. Yeah, well, it's impossible without that, right? But do you understand that logic? That this would have to be a Z minus makes enzymes all the time. So it has to be the operator.
Yeah.
That this one would have to be the repressor.
What
So,
were you saying earlier about the regulation of it, how it's unregulated?
oh, so if you get so normally the Lac Operon is called a regulated system, right? Because it's inducible. It's off in the absence of lactose and on in the presence. That's called a regulated system. If you mutate the operator so it can't bind repressor, you've now taken away the regulation, right? Because the only thing that's regulating it is the repressor. So now it's called an unregulated system. It's just on all the time. So anything that's unregulated and is on all the time is constitutively active. By definition, that's unregulated, right? Because there's no regulation at all.
Mm-hmm. Would you say that the love system is inducible as well?
Oh yeah, that's a good question. So,
the Lac Operon?
right. So this can be confusing, right? Whether you're talking about a repressible or an inducible system. The way to figure that out is don't think about the mode of regulation at all. Just, in other words, don't think about what protein is binding where, when. Think about the entire system. And basically, the way I always think about those is that an inducible system is off and it can be turned on, and a repressible system is on and it can be turned off. So if you look at the love system, right, when you look at the wild type strain 1, you see that when there's no love around, there's no enzymes around, right? And that you see in the presence of love, they're turned on. So that tells me it's normally off, but you can turn it on, which makes it just like the Lac Operon, an inducible system.
Yeah, that makes sense.
Yeah, so I think it's with transcriptional regulation, it's easy to get kind of bogged down in the details, but when you're talking about the system as a whole, that's all you're talking about. So you just say, is it normally off and turned on, or normally on and turned off?
Gotcha. Thank you so much.
Yeah. Sure.
How do you determine whether the enzyme is active or inactive?
For the purposes of this question, it's the same thing. So what that basically is designed because mutations can do two things, right? They can stop a gene being expressed, so it's just not seen, or it can make it inactive. So the protein is made, it just doesn't work. Phenotypically, those are the same thing. So it just depends on the type of mutation. So in this question, I take, either one of those answers is essentially gets the same credit, because you don't know unless you know the specific molecular version of that mutation. Right, so does anybody have any other questions? All right, well, if not, I know that was long, but thank you guys. I'm hoping that will be useful. I did mean to record this whole thing and post it, but I forgot to press record until about halfway through. But I will post what's left of it, at least, which is the last kind of half, in case you guys want to look at that. And I will put these notes on Moodle in just a minute. if you guys have any other questions before the exam, feel free to email me and ask those. And I will email you guys in the next day or so with, a few more details about the exam itself. So you know when it will be available and how long you will have to take it. Well, thank you guys. It's really nice to see you guys' faces, and hang in there. There's only a few more weeks of the semester, and let's get through it. And then hopefully in the fall, we'll be back on campus in some way, shape, or form. All right, guys, well, take it easy. Any questions, send me emails, and I will talk to you guys soon.
Thank you.
Thank you.
Have a good day.
Thank you.
All right, guys.
Thank
bye.
you.